And then what do we have instead of a serosa? The skin, okay? And then we move into the esophagus. We continue with the non-cornified stratified squamous epithelia. In the esophagus, remember, we do have a transition. We start out with the skeletal muscle, but in the middle part of the esophagus, we start to bring in the smooth muscle. In addition to bringing in smooth muscle in the muscularis externa, what do we add to the mucosa? Whenever we have the intercircular outer longitudinal smooth muscle in our GPAT, we also see a layer of smooth muscle at the deepest part of the mucosa. Remember what that was called? Instead of a muscularis externa, the muscularis mucosa. So in our histology model in lab, there was a few layers of smooth muscle right at the deepest layer of the mucosa before we transition into the submucosa. Okay? So the middle third of the esophagus, we pick up smooth muscle in the muscularis externa, we pick up muscularis mucosa, and as we move into the lower third, we leave behind entirely the skeletal muscle, and we have our intercircular outer longitudinal plan. What else is different about the esophagus? Do we have a serosa yet? No, not until the esophagus actually passes through the diaphragm, and we see the attachment, the cardiac, to the stomach, and it picks up peritoneum there. So what do we call that outer layer? The adventitia. Okay, same as the term that we use for the outer layer of a blood vessel. All right, we had one specialized modification in the esophagus at one region, the most distal 1.5 inches or 2 inches, that same area that's actually in the abdominal cavity had a modification. What layer of the esophagus was modified? In addition to having a serosa, there's one of the other 3 layers was modified because of its proximity to the stomach.
Muscularis externa at the...
Well, we have the cardiac sphincter there, yes. But I'm thinking of the submucosal glands, in the mucus glands, in the submucosa that send ducts up to the surface to protect the non-cornified stratified squamous from backsplash of acid. So those submucosal glands we don't see anywhere else in the esophagus, just at that last portion. All right, then we moved into the stomach, and we finished up lecture on Wednesday by talking about the different regions, and we looked at that in lab. So now I want to come back to the mucosa, submucosa, the general plan. And before I cover that for the stomach, we're going to be looking at the stomach, the small intestine, and the large intestine. And each of those has unique arrangements of glands, gastric glands and intestinal glands. All right. And so I want to just kind of diagram the schematic of that over on the far right corner, and then of this board, and then I'll refer to that as we go through those organs. So if we look at the mucosa of the stomach, and we'll come back and look at the detail of each of these. The surface epithelium forms gastric glands, but at the surface there are wide gastric pits. So this is identified as a gastric pit, and this is identified as a gastric gland. And like our standard pattern, the depth, these glands extend down to the bottom layer, and we have our muscularis mucosa. Okay? So that would be the mucosal pattern for the stomach. The mucosal pattern for the small intestine has intestinal glands. This would be like walking around on a barren landscape and having wide gopher holes. Those would be the gastric pits scattered around. All right, but nothing else, nothing growing up out of the surface. The small intestine has a greater need for surface area because this is where we actually do secrete most of our chemical digestive components and absorb the nutrient breakdown products. And so as we walk around, we have these gopher holes, but they're a little smaller and more of them. And we also see mounds like anthills. So the surface is thrown up into folds as well as wells being dug down in the surface. And so this would be the surface. This would be the lumen where the food is. So in the small intestine, we have these upward folds as well as glands. And these are intestinal villi. The single one is a villus. And again, intestinal glands. And an underlying muscularis mucosa. So we've added to our surface area because this level right here is equivalent to this level here. So we've increased the surface area. And then I'm going to put the colon over here just to match up with it. I'm going to move this back because I can eventually slide this back over. So then for the colon and rectum, if you're not yet aware, colon is another word for our large intestine. Okay? So by the time our food gets to the colon, our chemical digestion has basically occurred. We have cellulose left from the plants that we've eaten, and bacteria in the colon will break that down. So essentially what we do in the large intestine is reabsorb water. And if there's some fat-soluble nutrients, we might reabsorb those as well, including the vitamin K produced by bacteria in the colon. So we don't, are not necessarily that interested in increased surface area or absorption as much as we have just rows after row of intestinal glands. We don't have the pits. And as we'll get to that, those have a lot of goblet cells. Okay? So that's the pattern, the schema, if you will, for the mucosa of each of these three parts of the digestive tract yet to follow. But I'll just leave that there. Come back over here. So now we have the GPAT of the stomach. And the first component is the mucosa. And the mucosa, remember, is a mucous epithelium, a mucous membrane, so we have an epithelium. And from now until we get to the anal canal, it's always going to be simple columnar. Okay? And we only have two epithelia in our digestive tract. So mouth, pharynx, esophagus is going to be non-cornified, stratified squamous. And then at the stomach, there's an abrupt change to simple columnar. We see simple columnar all the way through the rectum until we get to the last 2 inches of the digestive tract, the anal canal, and we switch back to non-cornified, stratified squamous. It's pretty easy trying to keep those epithelia straight as compared to the respiratory system. So simple columnar. We do secrete mucus. We have mucus cells, but not goblet cells. We have a loose, irregular connective tissue. And we have our muscularis mucosa. So we're going to see this pattern for the mucosal membrane all the way through the rectum. And again, I'll draw a little bit larger our mucosa. So we have these gastric pits. And simple columnar epithelium. Just suggest it here as we go through. So for about this first portion here, the gastric pits, down to about this level, the cells are, when you look at them in the microscope, are a pinkish white color. They're mucus secreting cells, but they're not goblet cells. So they're called surface and neck mucus cells. And that protects the epithelia of the stomach from the hydrochloric acid that's going to be secreted up through the glands, as well as from digestive enzymes. Because our enzymes that we use in digestion break down proteins, and they break down lipids, increase the rate of breakdown, and break down carbohydrates, which is exactly what our cells are made up of. So the body is protected by a rapid mitotic. These cells replace themselves every 1 to 3 days. But even so, we need this mucus protection, all right? Then scattered among the cells of the intestinal glands, we have pyramidal shaped cells that stain pink to red because of their acidic nature. And these are parietal cells. And their role is to secrete hydrochloric acids. In addition to the parietal cells, most of the rest of the cells are a typical columnar secretory cell that are responsible for secreting digestive enzymes. And these are known as chief cells. So they're going to make enzymes. We do release some chemical enzymes in the stomach, though they're not of high importance. And then down at the base of the gastric glands, these cells have your nuclei up near the top because they actually secrete out the bottom of the cell. And these are endocrine cells known as G cells because they make a hormone known as gastrin that helps to regulate stomach activity. And then obviously on the bottom, we would have our muscularis mucosa. And this helps to throw the lining of the stomach into those longitudinal folds. Anybody remember what those are called? Rugae. Okay. We're going to see that term rugae 2 more times. We have rugae in the urinary bladder that we'll talk about on Wednesday, and we have rugae in the vagina that we'll talk about a week from Wednesday, on our last day of lecture. Okay, so that's the mucosa. Submucosa is standard. When I say standard, that means it fits our template. So it's our standard submucosa. It's irregular connective tissue, nothing remarkable. We find isolated lymphatic follicles and a little bit arterioles and small arteries and veins and lymphatics and so on. Okay. Then we get to the muscularis externa. And here we do find a modification. If you remember from lab, we have 3 layers of smooth muscle when our template was 2. We don't modify those other 2, but we add an oblique layer. So we add an innermost oblique, and then we have an inner circular and an outer longitudinal. And then finally we have our 4th layer, and here we truly have a serosa. What's the other name for a serosa? Visceral peritoneum. Which is a serous membrane, simple squamous epithelia on the outside attached by loose, irregular connective tissue. the major function of the stomach? Mechanical digestion. Okay? So this is not breaking any chemical bonds, though we do that a little bit, but this is the main function. So we started out in the mouth with some chemical enzymes, some enzymes that start to break down, and this is not a physiology class, so I'm just kind of giving you an overview. In the mouth, we start to break down carbohydrates. All right? If you chew dry crackers for 5 minutes or so, they'll start to taste sweet. But we don't get any product that we can absorb across the epithelium. Sucrose is too large to cross the plasma membrane. We also have lipase, but that does hardly anything. The lipid droplets are too large. We get to the stomach and all of that stops because the pH of the mouth is around 6 to 8, and those enzymes are denatured in the hydrochloric acid of the stomach. So whatever we started in the mouth no longer continues in the stomach. All right? As far as absorption occurs in the stomach, very little water is absorbed. You don't have the water channels there. Alcohol definitely is, because alcohol is soluble across the plasma membrane, and certain drugs. Okay? But again, for most intents and purposes, we're trying to create an increased surface area, and even the lipid droplets are broken down into smaller lipid droplets as we move that into the small intestine. All right, so now we're ready to move to the small intestine. The mesentery is associated with the stomach, where the lesser omentum and the greater omentum. What is the mesentery associated with the small intestine called? Mesentery proper, remember it's gathered up together and to ruffles that are attached kind of at an angle to the posterior body wall known as the root of the mesentery. So a little bit before we get into the mucosa, the GPAD of the small intestine, we have 3 components. The first component is a C-shaped structure which begins at the pyloric sphincter, where that was an increase in the smooth muscle. So this would be our pyloric sphincter. This C-shaped structure is about 10 to 11 inches long, almost a foot, and it's identified as the duodenum, or as I pronounce it, duodenum. You can use either pronunciation. And it's essentially a receptacle. Okay? It's going to receive from the stomach acidic kind, about 1 to 2 tablespoons of this churned food that has been processed in the stomach. Its pH being approximately 1 to 2. It's going to receive from the liver, which we will look at a little bit later today, bile salts. Those are stored in the gallbladder, so it may come directly from the liver, or it may come from a storage in the gallbladder. And then coming from the pancreas, we add bicarbonate ions, which neutralize the acidic pH of the stomach to a pH of approximately 8 to 9. I don't know why we have such a drastic change, because now the enzymes that the stomach makes, and it does release some enzymes that break down proteins and lipids, increase the rate of their breakdown, but they no longer work in the small intestine, because they need a pH of about 1 to 2 to work, and with the bicarbonate ions, the pancreatic enzymes and the intestinal enzymes need an alkaline pH. And so stomach enzymes no longer work. But these are added into the duodenum, and it's so short, there really is no measurable chemical digestion that takes place there. It's just a mixing and modifying of the pH. The duodenum is our first retroperitoneal structure. Okay, so it's been fused to the posterior abdominal wall and has peritoneum just on its anterior surface. At the end of the duodenum, right about where we see the superior mesenteric artery and the superior mesenteric vein cross over from behind the stomach. Just after that occurs, the intestine makes a right angle bend and comes into the peritoneum, and you can kind of see a fold there. This is now the jejunum. And it's about 8 to 9 feet in length, and it is intra. Peritoneum. In lab, most of our models, the rest of the coils of the intestine look like a bunch of snakes all coiled around each other. So this is the easiest, most significant way to say where the jejunum is located. It's at this bend, right after the duodenum. Okay? And hence that's where I will tag it. The jejunum is where we do a lot of our chemical breakdown. All right, the pancreatic enzymes increase the rate of breakdown, and we start to do some absorption there. And we do, another probably, we're at about 10 feet now. And then we finally, so dotted line, back and forth, and finally we get to the ileum, which is going to end by joining the large intestine. So the ileum is about 9 to 10 feet. It is also intraperitoneal, and it's where we do most of our absorption. Okay, so we finish digestion here and do most of our absorption of the food that's broken down.
Question, when
Yes.
you say, when we do where we do most absorption is in the ileum, that's the nutrients absorption.
Right, not the water. Water would be in the colon. But the simple sugars and amino acids and lipids. All right, some specializations. Actually, I'll do the specializations when we get to the mucosa. Before we look at the GPAT, just a little bit of physiology. When I talk about breaking down, enzymes don't really do the breaking down. They increase the rate of reaction. All right. When we talk about digestion, we use the term hydrolysis. Hydrolysis, if we take the words and look at the meanings, means the splitting of water, or we could say the splitting by water. For example, we have a carbon-carbon bond between our amino acids, between our simple sugars, between our fatty acids. Fatty acid is a long chain of carbons with COOH at the end and a bunch of hydrogens and so on. So we add with water, and what we get are these two components. So water actually is what breaks the bond. Enzymes increase the rate of that reaction, because if we didn't have the enzymes, and I've actually seen cool videos, time-lapse videos, where a sterilized piece of meat, so you couldn't assume that it was bacteria on the meat that was doing the breakdown, was suspended in a jar of sterile water, and it took a week of time-lapse photography to start to see the surface area of this break down with the water hydrolysis. Well, you know, we'd start starving by that time. So having the enzymes increasing, and they're specific to their substrate, so some enzymes will only work on a disaccharide, some will only work on a polysaccharide, some will only work on the amine side of a protein, some will only work on the carboxyl, the acid end of a protein, and so they can be pretty specific. Okay, but we have, so this is really the digestive process. Our enzymes are just increasing the rate of that reaction. Okay, so now let's look at the mucosa. And we really don't have to do a whole lot because this is our template. Template that we laid down on Wednesday was really, and the model that we saw in lab, was really that of the small intestine. So I'm only going to be adding a few specializations to different regions of the small intestine after I remind you of what this is. So mucosa. Our epithelium, again, is going to be simple columnar epithelia. This time we do have goblet cells. Again, the mucus secretion is not for trapping dust particles. We don't have cilia, we have microvilli. Increased surface area. In fact, we'll talk about 3 different sets of folding that increases surface area. We have our loose, irregular connective tissue, our lamina propria, and we have our muscularis mucosa. So now as I draw that larger, we have an intestinal villus. Formed of simple columnar epithelium with goblet cells. And microvilli. Again, this structure is the villus. And it's evaginated into deep intestinal glands. Which are also simple columnar epithelium. So these cells will migrate up onto the surface of the villus. It is here in the villus that we find one of the modifications that we didn't have a lymphatic lecture, so I probably didn't get to talk about this too much. But lymphatic capillaries, remember, are blind ending, whereas our blood capillaries are continuous. And so this lymphatic capillary has a unique name, a lacteal. So in a lab quiz or lab exam, if you identify it as a lymphatic capillary, that's true, it is, and that would be partial credit. But the best name for this specific location, and you can see it on our intestinal models, is a lacteal. It is significant because this is, like our other lymphatic capillaries, going to take up extra interstitial fluid, white blood cells, pathogens that are in the area. But in this location, it also takes up lipoproteins. So when we break down lipids as an oil, they're not soluble in the plasma of the blood, the water, because they're a non-polar covalent charge. Water is polar. So to get them to be soluble, we have to coat them in a protein coat that is polar. Well, by the time they get that protein coat, it's difficult for them to get into the blood capillaries. And so the amino acids and the simple sugars will go into the blood capillaries. monosaccharides, whereas our lipoproteins will go into the lacteal. Now, if we take a moment here, what happens to the capillaries of the small intestine? They form into venules, and what would be the vein carrying blood away from the small intestine? Which one? No, small intestine. So these are going to go, the capillaries are going to go into the superior mesenteric vein, and that's going to go into the superior mesenteric vein joins with splenic vein to form the hepatic portal. And that goes into the liver. So a hepatic portal vein is carrying blood that is rich in amino acids and simple sugars. The lacteal is going to go into the cisterna chyli, into the thoracic duct, into the subclavian vein, into the brachiocephalic vein, into the superior vena cava, into the heart. So how is it going to get to the liver? Without writing the whole pathway out? Hepatic artery. So it's eventually going to get into the aorta, and then it's going to get to the liver via hepatic artery. We'll visit this in a bit when we get to the liver's blood supply. So the hepatic artery, remember I talked about in lecture with the cardiovascular system, that the portal vein and hepatic artery are unusual, that they're both carrying parallel blood flow. Normally we have vein and artery going in opposite directions through into an organ and out of an organ. But at the liver we have the hepatic artery and the portal vein traveling together. One is oxygen rich and lipid rich. The other is oxygen poor but nutrient rich in amino acids and simple sugars. It takes both those vessels to carry what fetal vessel? What fetal vessel accomplishes the role of both of these? What's carrying oxygen and nutrients from the fetus to the liver? Umbilical vein. Okay? So this is our replacement for the role of the umbilical vein to get those nutrients to the liver. All right, so that's our mucosa. Our submucosa is standard except in two locations. In the duodenum, we have submucosal glands. And again, remember the duodenum is carrying acidic chyme. So the presence of these submucosal glands helps to protect the lining of the duodenum from that acidic pH until the bicarbonate ions have a chance to neutralize the acidic pH. All right? So those are present only in the duodenum, not in the jejunum or the ileum. And then in the ileum, we have a high number of lymphatic follicles, bunched very closely together, known as Peyer's patches. These are not lymph nodes, but bare naked spheres of lymphocytes. Okay, so dense area of lymphatic follicles. And that's there because the ileum is the last region of the small intestine before we get to the colon. And the colon has a normal bacteria, flora, the E. coli, and we don't want those moving back into the ileum and getting absorbed, crossing the epithelial membrane. And so we have a lot of Peyer's patches in that, a lot of lymphatic follicles in that distal area just before the ileum joins to the ascending colon. All right, the muscularis externa is standard, nothing different. And our serosa is standard as well. Again, this was the template that we utilized. Now, before we leave the small intestine, because of its function in absorption, secretion of enzymes, and absorption of nutrients, we have a high increase in the amount of surface area. Surface area is about the size of tennis court, two full set of tennis courts, with all of the membrane folds. May I erase this list of vessels here? So the folds occur at 3 levels. The smallest folds are microvilli. Okay, those are folds of plasma membrane. And then we have the microvilli, the cells of the microvilli surface on the fold of mucosa. And so the next fold is a villus. We're going to be plural. And so that is a fold. To make this plural. Fold of mucosa. And then, if we contract the longitudinal muscle slightly and the muscularis mucosa, this is actually visible to the naked eye without microscopes. You can look on the inside of the small intestine, and you see longitudinal, circular rings rather, of the submucosa. So when we look at a cross section here, then we would have our microvilli, our villi rather, on that. So this would be the muscularis mucosa, would be that black line right there. So the little villi would be sticking out from that. And so this is known as plicae circularis. Plica circularis would be singular. Those are the circular folds. Of submucosa. Plicae means fold. So if you were mailing pictures, and I don't know who does that anymore when you can send it by email, but if you were mailing pictures and you wanted to write on there, do not fold, you could put do not plicate. Nobody in the post office would probably know what you were talking about, but that's what it means, okay, or circular folds. Okay, so our pH of the small intestine is alkaline, about 8 to 9, and that's because of the pancreatic digestive enzymes, and we'll talk about pancreas towards the end of lecture. And so we've broken down our nutrients into amino acids and simple sugars and lipids, and absorbed most of them across the wall of the distal jejunum and the ileum. And now we're left with food that we haven't had time to digest, and we don't have the enzymes to digest, like the cellulose and the fibers of plants. And so that moves now, we now move into the large intestine or the colon. That's about 4 feet long compared to the 20 to 21 feet of the small intestine. And it's essentially the site for the absorption of water with some nutrients, but primarily water. May I erase this board? You guys all right with this? All right, so the large intestine colon. Starts on the right side of the body. Remember how the superior mesenteric artery was oriented over to the right. The ileum enters into it just above a region known as the cecum. Attached to the cecum is the vermiform appendix. Which has its own mesentery, so that means it is intraperitoneal. The appendix, like the Peyer's passage, has a lot of lymphatic follicles in its wall. And so for a long time, it still is considered to be an unused vestigial remnant of immune tissue, kind of like the tonsils. About 3 years ago, a paper came out in England suggesting, I don't know how you would prove it, that the purpose of the appendix is to function as a reservoir. So if you had a high level of dysentery, diarrhea, and you lost your bacterial normal flora, that the appendix could serve as a replacement, kind of a little incubator to replace those bacteria with. Well, obviously, I mean, I suppose that is true, but you're born without bacteria in your large intestine, and you get it as an infant, so the same thing is still possible. I suppose it could happen more rapidly with the appendix. But it is such a danger of getting infected and gangrenous or rupture that if any physician, if you're having abdominal surgery for any reason, they'll ask permission to go ahead and take it out while they're in there. The ileum kind of comes in at an angle, and this creates another sphincter known as the iliocecal sphincter, or more commonly, because it's more of an angle than it is an actual constricted muscle, sphincter muscle, it's also known as the iliosphincter, the ileocecal valve. Then we see the large intestine going up to the liver and forming a bend. So this is known as the ascending and this is retroperitoneal. This bend is known as the right colic flexure, or because it's close to the liver, the hepatic flexure. And there is mesentery that suspends it in position. We have the transverse colon. We have the left colic flexure or splenic flexure. And then we have the descending colon. Transverse colon is intraperitoneal. Descending colon is back to retroperitoneal. And then it forms a double series of curvatures before it goes straight. And so this is the sigmoid colon, which is back to intra. Remember I said every other one? So appendix is intra, cecum ascending colon is retro, transverse is intra, descending is retro, sigmoid colon is intra again. As we'll see in a few moments with our GPAT, the muscularis externa, the outer longitudinal muscle, is limited to 3 bands. And these are slightly contracted. I'll draw one, the other 2 will be kind of on the back side. And so these three bands of longitudinally arranged muscle are known as Taenia coli. The longitudinal smooth muscle. Its tone and contraction creates these pouches, which are known as haustrum. Plural is haustra. And it varies from animal to animal, as well as the speed at which fluid passes through here. So cows should have big fluid patties, and horses are drier, and rabbits have tiny little pellets, and that kind of thing partially is determined by these haustra and the rate at which fluid passes. The straight portion is the rectum. We'll draw that a little bit larger in just a few minutes. So, since most of the function, most of our fluid, our water, is absorbed across the colon, if someone has to have part of their colon removed temporarily, if they have ulcerative colitis or cancer, either temporarily while they allow it to rest or permanently, depending on where that colon is removed, and they can bring it to the surface and an opening in the skin called a colostomy, and they use an adhesive to glue a square in the skin that has an opening in it, and then they can change the bag by changing the attachment to that square that's already glued to the skin. If that colostomy is close to the proximal, remember our terms proximal and distal? Proximal origins the mouth. All right, so this part of the large intestine would be proximal to this region. So the more proximal the colostomy, the more liquid the fecal contents. So someone has a colostomy in the ascending colon, they would have a constant drainage of foul-smelling fecal matter. If the colostomy is done in the descending colon, then they often can be train themselves to have one bowel movement a day, like they normally would. And they would still have the colostomy bag in case of accidents, but it's dry, they can clean it, and there's not the bad odor and gases that there are with the more proximal colostomy. When I was a graduate student at Davis, I still had my RN, and I was working weekends at Center Davis. And I got a phone call from a friend she was a county home health nurse, and had a client who lived in Davis that needed a nurse to come over as the schedule worked out, it ended up about every 2 to 3 weeks. When he was 5 years old, he lived in Central Valley area, and had gone out and eaten lima beans, which were sprayed with some kind of pesticide. His mouth and esophagus and small intestine recovered, but his large intestine, the pesticide chemically burned away his mucus cells and epithelia and just left adhesions. So from the age of 5, he could no longer have a normal bowel movement. He had to have high colonic enemas. So he came up here pretty often for business. In his home in the Central Valley, he actually had a special bathroom built. And he had gone into the Army, took care of the animals with cavalry and stuff during World War II, came back, ran a business, and had an RN or a home health nurse come in. Most of us have a bowel movement every 1 to 3 days, okay? He got busy. He would go maybe 2 or 3 weeks. Can you imagine not having a bowel movement for that long? You start to reabsorb the toxins. And so it got to be the point where he knew he needed to have a treatment. He'd give me a call. And the first night I was over at his house, I was there from about 6:30 to 11:30. And he had a glass jar that would hold about 2.5 quarts, like a blender, without the blade at the bottom. A latex tube that we'd stretch to fill the bottom, and the tube itself was the diameter of my thumb. And I would give him anywhere from 2 to 4 quarts at a time, about 12 times. I divided into thirds, just regular tap water with ivory soap, kind of a soap suds enema, and then a natural saline. I'd add salt to the water. And his wife would be busy puttering in the back or painting or whatever. And I'd lubricate the tube. He always thought I did a great job because I was also an anatomist as well as a nurse, and I understood I just went slow. But you could feel the end of the tube and his transverse colon, and I'd see his toes start to curl, and encourage him to take as much as he could, and then he'd go sit in the bathroom. There's a bathroom off the bedroom for about 20 minutes, and I'd read my research papers, and he'd come back, and we'd do it all over again. And the first night, he pulled on his boxer shorts, came out, sat on the bed, and started to write me a check, and I just started chuckling, and he said, "What's the matter?" I said, "Do you know what this looks like?" So he was very careful to put RN after my name. And over the next couple of years, of course, we got to talk a lot, and I remember saying, why didn't you just have a colostomy? And he goes, "I'm so grateful that I never had to have that surgery," and he would handle it this way. His aunt, who was in her early 20s when he was 5, at the time already had to have high colonic enemas. She had been dating a guy, a dentist, and he was late for a dance that they were going to go to at the local fire station, and her friend said, "He knows where you're going to be, just join us." So she did. He showed up 20 minutes or 30 minutes later, and while they were dancing, he shot her in the back with a small pistol and paralyzed her. Now, depending on the level of damage to the spinal cord, many paraplegics can still they don't have feeling, but they can train their bowel movements and have normal bowel movements. But he had damaged the parasympathetic part, the distal part, the sacral part of her spinal cord. And so she already had to have those. And he relayed to me what a comfort it was to not be the only person that had to go through this. And so after he grew up, he actually took her into his home and took care of her. But it really brought home to me that part of his problem was not just the lack of the mucus cells we'll talk about in the GPAT, but all the adhesions as well. And both of those combined together, he just could not move. It just would stay stopped by the tissues, and as more and more water got absorbed, just become more and more blocked. So let's look at the GPAT. We have a little bit of modification here. Primarily in the mucosa and the muscularis externa. So if you notice the plan over there, the mucosa is just deep intestinal glands. Simple columnar epithelium. So nothing fancy except for the fact that 25% instead of about 5 to 10%. 25% of these simple columnar cells are goblet cells. So many that it looks like 80%. And here the mucus is not so much for protection against pH and digestive enzymes, but as I indicated with the story about my patient, for lubrication. Okay. Standard, everything else. Muscularis mucosa is present, and we have our lamina propria. We have our muscularis mucosa. Submucosa is standard. However, when we get to our muscularis externa, we have our inner circular layer, but the outer longitudinal layer is restricted to those Taenia coli. Okay. And that's kind of what's thought to form those haustra. Okay, the GPAT of the rectum is the same. And then we have the anal canal. So the rectum is going to have the intestinal glands and all of that. All right. The anal canal now, we move back to non-cornified stratified squamous. And we also add skeletal muscle. So the inner portion of the muscle around the end of the anus, and the anus is the opening of the anal canal. So this is smooth muscle, and it forms the internal anal sphincter. And this is skeletal muscle, and it forms the external anal sphincter. Now, our superior mesenteric artery and our vagus nerve provide blood supply and innervation to this left colic flexure. All right, so this is where superior mesenteric artery and vagus nerve supply stop. And vagus nerve. The blood supply for the descending colon, remember, is inferior mesenteric artery. And the nerve supply for parasympathetic, which is what activates our digestive tract, comes from that distal sacral spinal cord, that region that was damaged in that woman by her boyfriend's gunshot. And so that comes from the sacral spinal cord and is known as pelvic flank nerves. So no longer vagus. Because obviously that wasn't damaged when she was shot, because that comes from up here. So that's our last sphincter. So we have 5. We have the orbicularis oris for the mouth. We have the cardiac sphincter between esophagus and stomach. We have the pyloric sphincter between stomach and duodenum. We have the ileocecal valve or sphincter between small intestine and large intestine. And then finally at the end we have the internal anal and external anal sphincters. Now we have 2 major accessory organs to cover. Did I erase this board? You guys okay with that? And that's the liver and the pancreas. Until recently, with stem cell activity, you couldn't live without the pancreas. You still can't live without a liver. We'll look at the liver first. The liver, like the pancreas, is actually an outgrowth of the tube that forms your stomach and small intestine. So a pouch breathes outward. The liver, functional tissue of the liver is epithelial tissue. It's not classified as simple columnar or simple squamous because it's a gland. It's just glandular tissue. We don't classify it by layers. When we look at the liver from the front, we see 2 major lobes. We have the right lobe and the left lobe. It has a visceral peritoneum. If we turn the liver over and look at its posterior or inferior aspect, we still see these lobes, but we also see 2 smaller lobes. The inferior vena cava is wrapped around partially by the liver, so it helps to provide a good kind of marking point, all right? And we have our gallbladder kind of tucked here on that side. All the gallbladder does, by the way, is store the bile that the liver produces. So So in this region right here, and in this region right here, we see two smaller lobes of the liver. One is called the caudate and one is called the quadrate. Now, this is closer to your backbone, and a tail is posterior, caudal meaning tail. So that's how I remember that this lobe is the caudate lobe. Because it's closest to the posterior aspect. The quadrate is on the anterior margin, all right? And so this would be the quadrate lobe next to the gallbladder. Running down the mid-surface here, remember the peritoneal structure that I drew that came from the belly button, the umbilicus, and traveling in that fold was our collapsed ligamentum teres. So that's going to come in here. Well, it's the ligamentum teres and the collapsed umbilical vein. Anybody remember what this mesentery structure was? This fold of, double fold of peritoneum that suspended it, kind of like this? Falciform ligament, excellent. And then we have another component of the fetal blood supply, the collapsed ductus venosus, which you can see where it would have drained into the inferior vena cava, and this is ligamentum venosus. Now, in the center there, where we see the gallbladder, we also see where the portal vein enters into the liver and the hepatic artery. So this would be portal vein, hepatic artery, and then we see a third structure, which is the common bile duct. These three structures create what's known as the portal triad. Throughout the liver, we see these three structures always together. So, better than I can draw, I am going to put up a PowerPoint of the arrangement of the hepatocytes of the liver. We have a whole model in lab that shows these. So the liver epithelial cells are identified as hepatocytes, and the hepatic artery and portal vein come into the liver and branch within the liver. The branches go out and go up the sides of small liver cell groupings known as liver lobules. In humans, it's a little more difficult to see the six-sided arrangement, but in pigs, that's a little bit easier to see. And so, hepatic artery and portal vein will run up the corners of each of these structures, such as you see here. So each six sides of the hepatic lobule is going to have, let me just take one of these here, it's going to have this grouping. And so each corner is going to have a portal vein, hepatic artery, and it will share that corner with three other lobules. Okay, since they're six-sided, they come together. And along the sides of this hexagonal grouping, each will send out branches. And then the branches will join together and flow towards the center of the hepatic lobule. Think about a whole stack of bicycle wheels with the spokes running into the center, all right? Only the spokes are hollow and the center is hollow. So as the blood flows into the liver, it flows in through the portal vein and hepatic artery, which are flowing together. They join to form wide capillaries, which we call a sinusoid. The sinusoids now are mixed. Oxygenated, deoxygenated blood. The nutrients have been mixed, so now we're rich in lipids and amino acids and in simple sugars. And as they flow towards the center of this hepatic lobule, on each side is a row, a plate, if you will, if you look over on this side, a plate of hepatocytes. Every hepatocyte faces a sinusoid. So every single, they're no more than 2 cells thick. So if someone else stands on my back, I've got a sinusoid here I can dip into and take out nutrients. The cell behind me can dip into its own sinusoid. So every cell has access. And the liver is the largest gland within the body. It makes most of our plasma proteins, clotting proteins and transport proteins and osmotic proteins. All right. It also makes the bile salts that we'll talk about in just a moment that we added to the duodenum. Once the blood flows by the hepatocytes, it empties into a central vein. So in the center of each lobule is a central vein. And these central veins now are not completely depleted of nutrients, just the liver had first tap at it. Those central veins now come together to form the hepatic veins we described in the cardiovascular system, and the hepatic veins empty into the inferior vena cava. All right, so our blood flow is portal vein plus hepatic artery to sinusoid, to central vein, to hepatic vein, to inferior vena cava. So you already knew about the portal vein and the hepatic artery, and you already knew about the hepatic vein. So what we've added are the sinusoids and central veins in between. Okay. Now, the hepatocyte's basal membrane is not, here's one cell facing a sinusoid and the other cell facing a sinusoid, and two basement membranes come together, but not completely. There's a space right in the middle where they don't join, and this forms a tunnel. If I added a whole bunch of these short little tunnels up, so one cell is one tunnel, but I added 100, I create a long channel. And that's where the bile flows. So the bile is flowing in the opposite direction. The bile is flowing out towards these corners. And these tiny little channels are known as bile canaliculi. So here would be the sinusoid, and here would be the bile canaliculi flowing out. And at the corner, that portal triad we have is a bile duct. So now we're going to carry the bile duct out into the small intestine. So it's going in the opposite direction of our blood. So here's portal vein, hepatic artery joining together in the sinusoid. These would be the hepatocytes. So before we look at the pathway of the bile duct, here's another unique macrophage. Most of our macrophages are in the loose connective tissue. We had alveolar macrophages just under the surfactant in the alveoli. Here's a unique location of macrophages in the blood. And they are known as Kupffer cells. So that's another name for the macrophage. And their major function is to remove old, aged red blood cells. Red blood cells can't reproduce. They only have a lifespan of about 4 months. And their membrane gets fragile. And so the liver and the spleen are responsible for removing these aged red blood cells. Okay, so we'll talk about pancreas in lab, but before we leave, I want to diagram for you the pathway of bile flow. My screen's not going to go up, but I'll move over to the other side here, and hopefully you can see it from that side. So all the bile ducts come together, and bile canaliculi. So at each corner, we have a bile duct and a branch of hepatic artery. So this is kind of looking at the inferior aspect of the liver. So from the right side of the liver, and from the left side, we're kind of looking through the liver, we would have the right hepatic duct and the left hepatic duct, and the two join together to form a short, big surprise, common hepatic duct. Okay? Now, bile is being formed all the time by the liver. But we don't always have, well, some of us don't always have a high lipid content in our diet. And so if we're not eating a food that's rich in fat, most of that bile ends up being stored in the gallbladder. Our word for bladder is cyst. So this duct going to and from the gallbladder is known as the cystic duct. If we have a diet that's rich in lipids, the gallbladder will contract and send bile down the common bile duct. The common bile duct passes through the pancreas and it joins up with the pancreatic duct, and I'll have this diagram on the board. And then the two join together to empty into the small intestine. So this would be the main pancreatic duct. Where the two are joined is known as the hepatopancreatic ampulla. There's a smaller accessory pancreatic duct that empties here, but there's kind of a wide opening, not that large, known as the main or major duodenal papilla. And that's just kind of like the opening of the salivary gland duct in your mouth where this empties into it. Well, we can see that in lab. Thank you for your patience. There's not much on the pancreas, and we'll just cover that in lab as we go over the models.